everyone. I am Professor Charu Dogra Rawat from Ramjas College, University of Delhi. Continuing in our biotechnology series, today we are discussing about the molecular diagnostics for various genetic diseases. I explained about the molecular diagnosis based on the RFLP method, that how the restriction fragment length polymorphism can be employed to identify or diagnose various genetic disorders, how these RFLPs arise and how it can be used in a direct diagnostic method and in an indirect diagnostic method by employing the linked markers for the diseased genes to identify the diseases or to diagnose the diseases. To further explain it, I will use two examples, one of the cystic fibrosis disease and the second for the sickle cell anemia in this session to explain more about the RFLP based method of molecular diagnosis for genetic diseases. Now, cystic fibrosis, it is a hereditary genetic disease. It results in the accumulation of thick, sticky mucus which primarily affects the respiratory system, which is the lungs, the digestive system and other organs of the body. So, this particular picture shows that the cystic fibrosis occurs, can affect various organs of the body. Primarily it affects the lungs, where you can see this is a normal pathway, this is the normal passage which is there. But in the cystic fibrosis diseased individual, a lot of mucus accumulation is there in the epithelial layer of the lungs, the passage, and you can see that the air passing is obviously reduced because of the less space that they are getting. And this, not only this, but it also affects the epithelial layer of other organs, creating a lot of mucus. But primarily the individuals die, they do not go beyond 30 years of age, and they die because of the respiratory system failure. It is an autosomal recessive genetic disorder. So, I hope you understand autosomal, that the gene causing this particular disease is present on the autosomes. It is a recessive genetic disorder which tells that it is inherited in a recessive manner, that is, the disease will be manifested in a recessive homozygous condition. The dominant will be undiseased and the heterozygote will be a carrier for that particular disease. Cystic fibrosis is most common in the northern European populations. Approximately 1 in 25 Caucasians are carriers, that is, they are heterozygotes for the gene, and one child in every 2500 births is affected with cystic fibrosis. So, this picture depicts how the recessive gene is inherited in a Mendelian order. So, this is a very common inheritance pattern whenever we talk about the Mendelian inheritance. So, you can see that the two parents are heterozygotes. So, they are the carriers for that particular, in this case for the cystic fibrosis gene. Heterozygosis means that one allele will be the normal one and the other will be the diseased one, and then they pass on it to the progeny. So, in the progeny 25% will be both the normal alleles would be there. So, that will be the non-diseased state. 50% would be the heterozygotes and they will be the carriers. So, in this, one will be the wild type or the normal and the other will be the diseased one, and 25% will carry the disease because they would carry both the mutated alleles of the gene and therefore will manifest a particular disease. So, this is a general pattern for a recessive genetic disorder, and this is basically followed for the cystic fibrosis as well as sickle cell anemia as well. Now, mutations in the CFTR gene is responsible for the occurrence of the cystic fibrosis disease. This gene is located on chromosome number 7q31.2. So, you remember again how to identify these particular regions. So, in the 7th autosome, the 7th chromosome, non-sex chromosome, Q arm, in the region 3 and the subregion 1.2, this CFTR gene is located. The CFTR gene is basically a cystic fibrosis transmembrane conductance regulator. Transmembrane conductance regulator. So, it is present in the plasma membrane of the epithelial cells, and this gene translates into a protein that is a transmembrane conductor and basically a conductance regulator. The mutation in this particular gene affects the conductance of ions across the epithelial membrane and therefore manifests the disease. The CFTR gene encodes a protein that functions as a chloride ion channel and plays a crucial role in regulating the movement of salt and water across the epithelial cells. So, again you can see that how the gene mutation results in the cystic fibrosis. This is a normal individual in which this is the CFTR protein, and this facilitates the movement of the chloride ions into the cell. Now, when this is defective, now different types of mutations we will talk about. Different types of mutations result in different outcomes. Some mutations can result in the inactive, in the functional aspect of this particular CFTR gene. So, as you can see that the transmembrane protein is there, but the regulatory function, the conductance function is impaired. So, certain mutations can result in non-opening of this thing, or it is also termed as door jamming. So, in this case the chloride ions cannot move inside. You can see the thick layer of mucus that has accumulated on the epithelial cells, and therefore when this conductance is impaired, then it leads to the disease, symptoms of disease, and the person basically dies then from the respiratory failure. And then certain mutations, this is a very common mutation, and in this case the two-step mutation is there, which basically impairs the function as well as the structure. So, you can see this CFTR protein, it is formed, but it does not come and reside in the plasma membrane. It just keeps circulating in here. So, the structure is also altered, and because of that obviously the function is impaired. It is a double mechanism which is happening and causing the cystic fibrosis disease. You can see the thick layer of the mucus, and again the movement of the chloride ions is impaired in here, and therefore the CFTR gene is formed, but it is not able to locate itself. So, this is a impairment of location and function as well. This is just the impairment of function, but whatever the mutation is, it is causing the impairment in the flow of the chloride ions, which is very, very essential. The accumulation of the mucus is there, which narrows the passage for the flow of the air freely, and therefore causes the particular disease. Most common mutation is the delta F08 mutation, which results from a deletion of 3 nucleotides, leading to the loss of phenylalanine at a position 508 in the CFTR protein. So, let us look at what kind of mutations are present to cause the dysfunctioning, not functioning or location impairment of the CFTR protein, leading to the CF disease. So, first understand how the disease is manifested, and then we will look at that how this disease can be then diagnosed. So, the most common mutation is the delta F08 mutation, F508 mutation. So, in this the phenylalanine, so the mutations are named in this manner. 508 is the position, F is the phenylalanine, and delta basically means that it is removed, it is deleted. So, when this is there, this kind of impairment in the CFTR protein occurs, and this leads to both the location and function impairment that causes the cystic fibrosis gene. So, this you can see, this is the chromosome number 7, and in chromosome number 7, this is a particular sequence that is of the CFTR gene. So, this is the position Q31.2 as we said. So, in this particular sequence, you can see this triplet codon codes for the phenylalanine. This is the one, the 3 are coding for the isoleucine, and then the next 3, these 3 CTT are coding for the phenylalanine, and then the next one is coding for the glycine. So, in the CFTR mutation, whenever the gene is mutated, these three, one codon from the isoleucine and the other two codons for the phenylalanine. But the continuation so occurs that in case of isoleucine, this A, T and this T, the black T, then becomes a particular codon, and because of the the third position wobble hypothesis, this ATT also codes for isoleucine. So, you get this continuation, but this phenylalanine which is there is basically removed. So, this is not present there. So, the removal of this particular gene is there, protein amino acid is there, sorry. So, other notable mutations include a G542X, which is a nonsense mutation. So, as the name suggests, it introduces a premature codon. So, the glycine at 542 position and X means that introduces a premature stop codon, and G551D. So, at position number 551, the amino acid glycine is replaced by amino acid D, which is the aspartic acid. So, that is how the mutations are named. So, a nonsense mutation, a missense mutation, missense mutation, this can also lead to the impairment in the CFTR protein. But as we discussed, this G542X would probably lead to the formation of the CFTR protein and its incorporation into the plasma membrane of the epithelial cells, but it will impair its function, or the GFF551D. So, different kinds of mutations can lead to different functions or impairments in the CFTR gene functioning, but it leads to the cystic fibrosis disease, both of them. So, now let us look at the diagnostic methods. As I said that the CFTR gene can be diagnosed by a direct diagnosis method provided the specific mutation is known, and it can be, if it is not known, but the gene we know, the CFTR gene, but we do not know what mutation is there, we can do an indirect diagnosis method as well. So, let us first look at the direct diagnosis method. So, in direct diagnosis method, for example, the G542X mutation, as I said, it abolishes a recognition site for the enzyme HINC2. So, G542X is basically the occurrence of a stop codon, and this abolishes an enzyme site which is HINC2. And because of the abolishment of a particular restriction site, there would be a restriction fragment length polymorphism. In the normal allele, HINC2 cleaves the PCR amplified DNA into two fragments. However, in the mutant allele, the enzyme cannot cut the DNA, resulting in a single larger fragment. So, that is basically how the difference in the fragment size will be observed when we run the agarose gel electrophoresis, and therefore, we can identify or diagnose that the CFTR is mutated. So, I hope it is clear that in the normal allele, the HINC2 is cleaving the PCR amplified DNA. So, when you run this, so you isolate the DNA, you PCR amplify it. For PCR amplification, the mutation need to be known, because you will use those primers to amplify that particular region. And when you observe two fragments, that means it is a normal allele. But when you obtain a single larger fragment, then it is a mutated allele. So, this is a direct diagnosis method for cystic fibrosis. Cystic fibrosis mutation delta F508 that we said, it is usually detected using PCR followed by size differentiation. So, again followed by size differentiation. So, what you will see here is, just look at that, if they are the normal alleles, then when you PCR amplify it, you will get a single band. And that single band, so you are not using a restriction fragment length polymorphism, because you are not restriction enzyme digesting the PCR DNA, but you just amplify the cystic fibrosis CFTR gene, and you will identify, or this particular region basically, and you can identify the different size of the fragments when you run it on the agarose gel. This is not, obviously as you can see, the size difference is just 3 base pairs. So, this is not identified on a normal agarose gel, but this can be identified on, for example, the polyacrylamide gel where resolution is too high, so that you can identify this 3 base pair change. So, in this double normal allele, you will have a 98 base pair fragment. In the carrier, you will have 2 fragments, one is a 95 base pair fragment and another is a 98 base pair fragment. And because of the deletion of the 3 base pairs, you will get a difference in the size of the 2 fragments. And if it is a diseased, that is, it is carrying both the alleles as the diseased mutated gene, then you will have a single fragment which will be 95 base pairs. So, this is a simple diagnostic method that can be employed to observe the size differentiation of the CFTR gene region containing this delta F508 mutation, and you can diagnose whether it is a carrier or whether it is diseased or whether it is a normal, both the alleles are normal or not. Then as I said, again this is a size differentiation method, but this delta F508 can also be identified in an indirect diagnosis method. And in this indirect diagnosis method, what we identify is a marker which is near to it. So, you can see this marker, this KM19, which is basically near to the CFTR gene, and we identify the polymorphism in this particular marker. The chromosome that lack the PST1. So, for example, this is a heterozygote carrier. These two are the carriers, the pedigree is shown here. So, when we show it like this half, which basically means that one is a normal allele and one is a diseased allele, so this is a carrier. And in this case, this complete darkened is the diseased one and the complete non-darkened is the non-diseased one. So, you can see that the carriers have the probability to have the homozygous recessive condition in which both the alleles are of the diseased state. It has the possibility of a non-diseased individual where both the normal alleles combine together, and therefore, you can have these kind of variations which are there. And if they had this kind of a progeny, then their DNA can be amplified, that particular region, and then they can be digested. So, in this case, the chromosome that lack the PST1 cutting site appear as a single 950 base pair band. However, the chromosomes that have the cutting site appear as the 650 and 300 base pair bands. So, you can see the difference in the size of the bands which is there. So, you can see again the genotype, it is shown here by plus and minus, that is which is carrying the marker restriction site and which is not carrying the restriction site. So, the one which is carrying the restriction site, for example, this is the diseased one, is basically showing the different sized, and the other one is showing the different sized fragments which are there. So, this differences in the size of the fragments, this 300 base pair is too small. So, sometimes it is not identified, but basically this differences in the 950 and 650 base pairs can be clearly identified in the diseased and in the diseased and in the non-diseased state. So, this PST1 site is abolished here, but the PST1 site is intact here. So, this is showing the particular disease or the mutation, and this is not showing the mutation. So, I hope this indirect diagnosis technique is also clear to you. The main thing about indirect diagnosis is that we do not look at or identify the inheritance of that particular gene or mutation, but we identify a marker which is linked to that gene and study its pattern of inheritance or the polymorphism that is occurring and predict diagnose that the presence of a particular disease is there or not. So, the cystic fibrosis method, as I said, if the mutation is known, one can employ the direct diagnosis method where we use the size differentiation to identify the normal allele and the diseased allele, and if the particular specific mutation is not known, then the indirect diagnosis can be done, so that a linked marker such as this KM19 or different markers are there, WC1 there is also, then those markers are basically identified and their inheritance is studied, and because it is linked to the CFTR gene, in the CFTR gene again this kind of pattern is shown. So, in the CFTR gene you can see there is a presence of a particular site, restriction enzyme site, and there is no presence of a restriction site here. So, if there is a presence of a restriction site, then you will get two different kinds of fragments, and if it is a normal gene, then you will not get a different, again you will get a different kind of a fragment. So, it depends upon whether the diagnostically you get one fragment or two different fragments or a smaller size fragment. The difference is in the size of the restriction fragments that you are getting. So, this is the indirect diagnosis method. Now, let us look at another disease which is called as the sickle cell anemia. Sickle cell anemia is again an autosomal recessive genetic disorder, autosomal present on the chromosome, autosomes, recessive again inherited in the same manner as cystic fibrosis, that is the homozygous recessive individual is going to manifest and show the disease. The homozygous dominant or the heterozygous will not show a particular disease. So, this is an autosomal recessive genetic disorder. The incidence is that it affects more than 100,000 people in the United States and 8 million people worldwide. 1 out of 400 African American children in the United States are affected, and even more than that, you know. So, it is a sickle cell anemia is a very widely spread disease globally, which is there. Genetically, there is a single nucleotide substitution where A is converted to T or is replaced by T. In the 6th codon, GAG gets converted to GTG of the beta globin gene. This mutation leads to the amino acid substitution of valine for glutamine at position 6 of the beta globin polypeptide. So, this alpha and beta globin, there are two chains, polypeptide chains which form up the hemoglobin, and that hemoglobin is basically responsible for the RBCs carrying the oxygen, the capacity. So, in this beta globin polypeptide at position 6, there is a change in substitution in one nucleotide that leads to the replacement of 1/6 codon from valine to glutamine, and that causes this particular disease. So, in this case you can see that this is a normal gene and this is a there is a point mutation that is occurring. So, there is a conversion, there are two polypeptides alpha and beta. So, these are the normal ones, and you can see there are normal shaped RBCs which are there, but in the sickle cell anemic case, the beta globin gene polypeptide alters because of the change in the amino acid, and during deoxygenation they polymerize and therefore give large stretch to the RBCs, and they become such kind of a sickle cell shaped, and obviously their oxygen carrying capacity is far reduced and the disease is manifested. There are various symptoms of the sickle cell anemia. As you can see, it is not only affecting the oxygen carrying capacity of the blood, but it is also impairing the functionality of many different organs. For example, the eyesight also becomes weak. There are infections that occur, and all different kinds of impairments are there which is associated with the sickle cell anemia which is there. Now, sickle cell anemia, one can do a direct diagnosis method. The mutation that causes sickle cell anemia coincidentally resides within the recognition site of certain restriction enzymes such as MST2, CVN1 and BSU2. Now, because these mutation lie within this restriction sites, so if the mutation occurs, then obviously the restriction fragment length will be differed, and that can be easily directly diagnosed on the by the gel electrophoresis. Individual with the sickle cell mutation lack the restriction site present in individuals with normal beta globin genes. So, recognition site is present in beta globin gene, but due to the mutation which resides in these restriction enzymes, that mutation is abolished, and therefore you will get a different fragment which is there. So, let us look at two of these restriction enzymes, that how they lead to the direct diagnosis. The first is the use of the southern blot hybridization. So, you can see this is a normal gene, and you can see if it is a mutated gene. So, this is the restriction site which is present here for the CVN1 and CVN1, and then you get a different mutated allele where this site is abolished. So, you transfer it, you run it, you digest it, you run it on a gel, and then you put it on the nitrocellulose membrane and then probe it, do the southern blot hybridization, And in the southern blot hybridization, you can see the different states. The one where there is no disease, the one which is a carrier. So, one of the allele is normal and one of the allele is diseased, and the one where you have a larger fragment where this site is demolished, therefore, you will have a larger fragment. So, you can see the difference in the restriction fragment lengths that can be used to easily diagnose that whether the individual is a carrier or is manifesting the sickle cell disease, or the fetus. It is basically at the fetal state also, one can predict whether it is there or not. This is again another example in which there is a PCR amplification which is there, and in this PCR amplification again you can see that these two are the carriers and these are the diseased state, non-diseased state and the carrier state. So, in non-diseased state you can observe these two fragments are present, but in the diseased state this larger fragment is present here. So, it is showing the gel running from that side. So, this is the larger fragment which is present there, and these are the two fragments, 140 base pairs and another one which is present in the non-diseased state. And if you look at the carrier profile, you will observe all the three bands are there. So, the one which is the larger one as well as the one which is the one which is present in the normal one. So, looking at the profile, you can observe the differences in the fragment length, restriction fragment lengths, and you can easily identify that easily diagnose whether it is a anemic person or it is a non-anemic person. This is again another example using the MST2 gene. So, again you can see here this is the sickle cell anemia gene that we are talking about, and you can look at the restriction enzyme sites which is there. This is the beta globin gene, this is the normal gene, and this is the sensitive gene which is present. So, in the sensitive gene, the beta globin sensitive basically means a sickle cell anemia gene. So, sickle cell anemia gene, there is an abolishment of A to T transfer is there. This is the GAG codon at the 6th position. This is the GTG codon, and because of this occurrence of a GTG, this this MST2 site is basically abolished, and because of that abolishing site, you can see that you get a larger fragment which is there in this case, and in the normal you get two smaller fragments which are there. So, if you have got a homozygous normal alleles, all you will get is these two fragments. So, you can see this pedigree as well. These two are the carrier parents. This is a non-diseased individual, this is a diseased individual, and this is a carrier individual. So, just compare the profile of these three individuals. So, in the non-diseased state, you can observe these two fragments are present because this MST2 site is intact, and as you can see the genotype as well, they are the normal hemoglobins which is there. In the diseased one, you get a larger fragment. So, this is the larger fragment where this particular restriction site is abolished, and in the carrier state there are three fragments which is present because there is a one normal allele giving rise to the larger fragment and two smaller fragments giving because of this presence of this particular allele. So, easily you can diagnose that whether the individual is a diseased one, a carrier one, or a non-diseased one. So, this is the molecular diagnosis of the cystic fibrosis and sickle cell anemia that we have discussed, and one method that we have discussed here is the restriction fragment length polymorphism based method. The direct diagnosis method where the occurrence of the polymorphism in restriction fragment lengths is due to the creation or abolishment of the restriction fragment recognition sites in the diseased genes which is there. And the indirect diagnosis method, when we do not know about the mutations of the gene or we do not even know the gene as well, then we identify the linked markers and based upon the RFLPs we diagnose the particular disease. In our subsequent sessions, we will talk about the other different methods such as the OLA method or the dot blot method to for the molecular diagnosis of genetic diseases. Thank you.